In this lecture, we're going to take a look at the concept of the receptive field. And the receptive field refers to that region of the skin where the depression of the skin or mechanical stimulation of the skin will discharge these sensory neurons underneath that particular stimulus. So in other words, the receptive field refers to the area of skin captured and sensitive by a particular neuron, but can be expanded to an entire nerve as well. And this is typically examined through an experimental setup. Here we see for a person in which a stimulus probe, this is just an external probe that can apply a very small amount of pressure or slightly more pressure over different parts of the skin through mechanical stimulation. And a recording electrode, in this case, is inserted into an accessible nerve, such as the median nerve. And the tracing here illustrates that when the stimulus probe is applied to the skin, that activity increases along that median nerve, confirming the receptive field detected by the neurons, sensory neurons in particular, that are part of the median nerve. On this slide here, what we see is an illustration of the receptive fields for both the superficial and deep layers of the skin in the upper and lower tracings respectively. So the slowly adapting mechanoreceptors have been identified through these Merkel disc receptors at these unique locations, and you'll notice that in the superficial layers, the receptive fields are much narrower than it is in the deep layers with the Ruffini endings. So the Ruffini endings and Merkel disc receptors are both slow adapting, which means that they're going to continue discharging action potentials with the application of the stimulus. But in the deep layer, we see a wider field, a wider surface area of the skin captured by these particular receptors. And these ones here in the deep skin are stimulated by skin stretch. In terms of the fast adapting or rapidly adapting mechanoreceptors in the surface layer, the Meissner's corpuscles are the neurons, the sensory neurons that have been found to cease discharge even with a sustained application. As illustrated in the previous video, the Pacinian corpuscle is also fast adapting, but because it's located deep in the skin, it's able to detect depressions or external contacts over a wider surface area of the skin. And in these tracings here, the small circle represents the point of maximal sensitivity for those receptors, although with the deep ones, we can see that the receptor is still sensitive, not to the same magnitude, but is still sensitive outside of that maximal sensitivity spot. To illustrate the receptive fields in human skin, this study published in 2001 sought to examine the influence of knee joint position and the speed of the movement of the knee joint on proprioception. And as part of this study, we won't focus on all of the results, but as part of this study, the researchers examined the receptive fields and attempted to map out the receptive field of the thigh. So 16 participants volunteered for this study. 11 were the data of 11 of those participants were able to be used. And they used a technique called microneurography, in which very small recording electrodes of 200 microns in diameter were inserted into the lateral cutaneous femoral nerve. Now, because the term is cutaneous, this is a sensory nerve that provides sensation to the lateral portion of the thigh. And experimentally, once the electrodes were placed into this nerve, the researchers passively moved the participant's knee into a slight degree of flexion from full extension. So moving into flexion from extension would actually stretch the skin region on the anterior lateral portion of the thigh. The researchers also applied electrical stimulation um, into the skin in order to help establish the receptive fields while simultaneously measuring the discharge of the sensory receptors, not just to the mechanical movement, but also to the electrical stimulation. In conclusion from this study, the researchers found that stretching the skin or skin strain was sufficient for activating mechanoreceptors, and they found that the slow adapting mechanoreceptors were the most important or most active during the knee movements. And they also determined that the hair follicle receptors were not involved in triggering or in sending signals to the central nervous system regarding knee motion. In this illustration here, the researchers attempted to map out the receptive field for the lateral femoral cutaneous nerve. And as you can see here, that the different participants in the study, and these drawings represent the different participants through a series of different experiments, that the receptive field did vary between people. It was consistent that the nerve provided uh, uh, sensory sensation to the anterior and lateral thigh, but the receptive field was quite a bit different, although they were all centered over that particular region of the thigh. In terms of the center of the receptive fields, once again here, you do see quite a bit of a range of these different types of receptors, including the slow adapting receptors, fast adapting, as well as low threshold C mechanoreceptors. So these are the ones that transmit slowly. But once you see here, the main point here is that there are a range of different receptive field centers that will cover the anterior and lateral thigh. The summary or the outcome of the study can be summarized in this graph, which did focus on the slow adapting receptors. And by referencing the knee movement here, the knee angle of 0 degrees indicated full knee extension. So the knee was completely straight, and then the researchers passively would move the knee up to 30 degrees of flexion. And that's illustrated in the second tracing, where the knee at rest at 0 or full extension, you can see that the knee was flexed, and recall that flexion would stretch the skin over the anterior lateral portion of the thigh. And so if we isolate this region here in the first experiment or the first movement here, so we see one person's leg was moved into a flex position, not quite to 30 degrees, but approximately 15 degrees. There was a corresponding increase in the discharge rate of the lateral femoral cutaneous nerve. And the magnitude of that discharge rate increased and paralleled the increase in the knee joint angle. So as the knee was flexed and the skin stretched, the impulses were transmitted, showing a very clear, close correspondence, and that's supported by the statistical measure here of agreement at 0.88. In terms of the effect of angular velocity, we see certain instances where the steepness of the knee angle joint with time increased at different points. And if we track the angular velocity, as you can see here, the angular velocity in this particular set of experiments increased periodically from a knee flexion back into extension, flexion back into extension. Now we see not just a greater amount of knee flexion up to 30 degrees, but the movement was faster, and we can see an even faster movement here. And so what we see here overall is that at these different regions of knee flexion, both in magnitude and speed, that the discharge rate followed that movement quite closely. And that at faster speeds, we tend to see a faster spike in the impulses. Likewise here, we see a very fast impulse. And even though the knee was held at that same position, so here we see the knee held at 30 degrees. In the most extreme case, the lateral femoral cutaneous nerve, even though the impulses, the nerves still accommodated, the impulses were sustained, which means that although they adapted very slowly and never returned back to the baseline until the leg was moved back into full extension, indicating that the sensory neurons embedded within the skin that are part of the lateral femoral cutaneous nerve were not just stretch sensitive, but were also velocity sensitive as well. So this ends our final video of part 1.